Hello everyone. In the last lecture, we have discussed about the fundamentals of living system, and particularly I mentioned that genetic material is one of the most important thing that should be present inside all the living unit. And now in this lecture, I am going to discuss about the discovery of genetic material. I will particularly explain three classic experiments which help us to realize that DNA is our genetic material. The first one is the discovery of transforming principle. So, this is very exciting and important experiment. So, like all of us know that in 1918 there was Spanish flu. So, during that time, like more than 100 million people died because of Spanish flu, and that is roughly about 5% of world population. And scientists across the world, they are trying to develop some kind of therapeutics. And during that time, it has been noticed that although Spanish flu, like it's a viral disease, but the thing is, because of that viral infection, the immune system get weak, and because of that, some secondary infection come. And during that time, Griffith, he was a British microbiologist, so he was working with Streptococcus pneumoniae. So, this is bacteria which causes pneumonia, and he was trying to develop some kind of vaccine against Streptococcus, so that the people can be cured from pneumonia. So, what he found that there were two types of Streptococcus pneumoniae. So, one is R type. So, R stands for here rough surface, something like this, rough surface. And another strain of bacteria, he found S type. So, surface is smooth surface. So, why they are smooth? Because they have very thick polysaccharide layer on the outer side, and they are the smooth bacteria. Now, this R strain of bacteria, they are non-virulent. Non-virulent means they cannot cause the disease. So, for example, this rough surface bacteria or non-virulent strain, if we inject that in some mouse, the mouse will survive, they will not die. So, this is called non-virulent, right? And now, the smooth surface bacteria, S type bacteria, so they are virulent. So, as a result of that, if we inject this type of bacteria, S strain of bacteria, so the mouse will die. Now, the thing is, why this smooth surface bacteria is virulent? Because of they have this polysaccharide coating outside, so they escape host immune system. So, as a result of that, host immune system cannot detect properly. As a result of that, those S strain of bacteria, they can multiply and they can cause the disease. As a result of that, mouse will die. So, now Griffith did a series of experiments, and he proved some interesting thing here. So, what happened? Griffith used mouse as model organism. So, now in this mouse, if we inject what Griffith did, so he injected R strain or the rough surface bacteria here, R strain bacteria. So as I already mentioned, so if we wait for some time, few days, the mouse will survive. That I already mentioned, because the R strain is non-virulent. So as a result of that, mouse will survive. Then in the next step of experiment, what he did, he injected S strain of bacteria into mouse. And now the mouse will die. So die, because the A strain is the virulent strain. This is plain and simple. But in the next interesting experiment, what he has done, he again injected S strain of bacteria, but it is heat killed. So that means he just boiled the bacteria, just like pressure cooker or some kind of autoclave. So he just boiled this bacteria. So this is called heat killed S strain. So since those bacteria, they are dead, so they cannot cause the disease. So as a result of that, again mouse survived. So now the most interesting experiments coming here. So what he did, he injected heat killed A strain bacteria plus live R strain of bacteria. So together he injected this. Now the result was surprising, because see, individually heat killed bacteria, they cannot cause the disease and mouse can survive. Similarly, only R strain, they cannot also cause the disease. But in this case, surprisingly, he found that mouse died. So this is very surprising at that time, because individually the heat killed S strain, it cannot cause the disease. At the same time, live R cells, that also cannot cause the disease. So he actually hypothesized that something is present in the heat killed S cells, and that converts R strain of bacteria into virulent strain, so that the mouse can die. Not only that, more importantly, when he took some sample from this dead mouse and culture in the laboratory, he found from this mouse, he found that smooth cells bacteria are appearing. Smooth cells bacteria, they are appearing in the culture plate. Not only that, this transformation, this changes is permanent. If you grow these cells again and again in petri plates, so they will give rise to smooth cell bacteria. So that means this change is permanent. And this is, he mentioned that this is transforming principle. This is very important, transforming principle. But what is transforming principle? What is the ingredient? That was not known during that time. But something is present in heat killed S strain that convert R cells into S cells. And unfortunately, he he didn't realize everything when those things happened later on, because this time whatever I mentioned, the experiment is 1928. Then soon after Second World War started, and in 1941 he passed away because of some bombing in his laboratory. So as a result of that, he didn't see the final conclusion of this experiment. Later on, some other scientists, they proved that this transforming principle is nothing but DNA, and that stored the information. So as a result of that in heat-killed S cells in S strain, although the bacteria is dead, but some of the DNA present in this strain, that went into the R cells, and it convert the R cells into S strain so that they can make now this polysaccharide layer on the surface. And as a result of that bacteria can survive, it can escape host immune system, and therefore it can cause the disease and it can kill the mouse. But all these things came later by some other group of scientists. So here I would like to mention that the experiment that proves DNA is our genetic material. Griffith just told this is transforming principle. It's very important, but a series of experiments conducted by Avery, MacLeod and McCarthy demonstrated that DNA is our genetic material. So briefly, I would like to mention here that what they did, Avery, MacLeod and McCarthy, so they took cell suspension, that means cell-free extract of S strain of bacteria. So cell-free extract of S strain of bacteria. So there is no cell, so only the extract of S strain. Then what they have done, so they were trying to do different types of experiments. So they are trying to fractionate it, so that from the cell extract they are removing carbohydrate, they are removing a lipid, sometimes they are removing protein or nucleic acid, and then they are trying to see what is the chemical nature of transforming principle. That means when you are removing the lipid and carbohydrate and protein, still it can transform this bacteria or not, transform this R strain to S strain or not, because they cleverly escape, or I would say they avoid the use of mouse here. It will take longer time. What they have done, they took this cell-free extract, and then if you add this extract in some tube where you have R strain of bacteria, right? So you are just putting this cell-free extract here, and if you culture this bacteria, R strain of bacteria, and after some time they observe that in this tube S cells bacteria also appeared. That means they avoid the use of mouse, but still you can tell that the cell-free extract of this S strain of bacteria, it can convert R strain into S strain. So now this is again S strain. Now what they are trying to do, they are trying to analyze what is the chemical nature of this transforming principle. So as a result of that, they took this cell extract and then they removed lipid and carbohydrates, by some biochemical method. So now their hypothesis is in this cell-free extract now they have proteins and nucleic acids. So, nucleic acids means here DNA and RNA. So, deoxyribonucleic acid and RNA ribonucleic acid. So, all these three things are present in this cell-free extract. Now, what they have done, so they treated this extract with some protein destroying enzyme. So, they used pepsin. Pepsin will destroy protein. So, they use pepsin. So, as a result of that, they would expect that there is no more protein. So, they have DNA and RNA or nucleic acids present there. Now, when they use this extract and again they put into a tube where they have R strain of bacteria, they found that this R strain can be transformed into S strain again. S strain again. So, that means protein is not responsible for this transformation. So, although they destroyed protein by adding pepsin, still it can transform R strain into S strain. Then what they have done, they also added in a different experiment, they add ribonuclease. So, ribonuclease, this is again another enzyme. So, it will destroy RNA. Then we have here proteins and DNA. And this one has the ability to convert R strain of bacteria into S strain. So, that means the transforming principle is still present here. So, therefore, they have concluded in 1944, they have concluded that DNA is responsible here because they have destroyed both proteins as well as RNA, but still it can transform R cell into S strain or smooth version of this bacteria. But we have to be careful here because during that time, like the scientific field was dominated by protein biochemists. Like almost everyone, they believe that protein, they do function for us, not the DNA, because DNA sometimes thought by scientists that very boring molecule, because DNA has only 4 nucleotides, its repetitive structure only. But proteins, on the other hand, they have 20 different amino acids and their side chains are so different and they can do function for us. So, as a result of that, it was very difficult for Avery, Magloyd and McCarthy to convince scientific community that this is DNA and DNA is the responsible for this transformation. But they also tried to carry out some more experiment. For example, they use some ultracentrifuge to and when they centrifuge this cell-free extract, they found that something coming at the bottom of the tube. So, that portion should be very high molecular weight, and only that bottom fraction of the ultracentrifuge tube can transform R strain into S strain. So, therefore, they again predicted that something very large polymer that is responsible for this transformation. So, for example, lipid, carbohydrate, protein, those are much smaller compared to long stretch of DNA. So, as a result of that, they are again concluding that DNA is responsible for this transformation. But the major problem was during that time that some enzyme which will specifically cleave, specifically destroy DNA was not discovered. In 1946, McCarthy, so he purified DNase, deoxyribonuclease enzyme, in 1946. And then he proved that whenever he is adding this enzyme DNase, then the transforming principle, it cannot, whatever present in this extract, if you add DNase, then it cannot convert R cells into S cells anymore. So therefore, it establishes that DNA is the genetic material. Whatever present, the information present in DNA, that actually transform the character of R cells into S cells, and they can produce a polysaccharide layer on the surface, and therefore they established, or this experiment proved, that DNA is our genetic material. But as I told you, so this is in 1944, this is Second World War time, and most of the scientists, they didn't pay that much of attention here. Part of the reason was, as I told, that during that time, most of the scientists believed protein is the ultimate thing. But again, another round of experiment done by another group of scientists, Hershey and Chase, they definitively proved that genes are made of DNA in 1952. So this is very exciting experiment, and after this experiment, everybody, like scientific community, they agree that DNA is our genetic material. Briefly, what Hershey and Chase they did, so they took some fudge virus, right? Fudge virus. So T2, this is bacteriophage. So bacteriophage means they will kill bacteria, bacteriophage. So T2, this is bacteriophage. So what happened? It infect bacteria, for example, E. coli. If this T2 infect E. coli, and then within 40 minutes or 1 hour, this virus will multiply inside E. coli and they will burst E. coli cell, and millions of T2 phage will be produced from this E. coli. Now, they utilize this system to prove that DNA is our genetic material. The beauty of this system is this T2 phage or virus, they don't have so many complex things inside their body or inside the virus. So they just have proteins, generally that form the coat of or surface of the virus, and they do have nucleic acid or DNA, proteins and nucleic acid. Now, what they did, they labeled T2, bacteriophage with radioactive phosphorus 32P. In some culture, they developed this radioactive label T2. This is 32P label T2. And now, in another experiment, they label this bacteriophage with radioactive sulfur, 35S, radioactive sulfur. So again, they got T2, but it's radioactive sulfur level. So this is sulfur and this is phosphorus radioactive. Now the thing is, as I already told before, that phosphorus is present in DNA or nucleic acid. So as a result of that, in this case, DNA should be radioactively labeled. So DNA should be labeled with 32P. And in this case, proteins should be labeled because proteins should be labeled with 35S or radioactive sulfur, because phosphorus is not present in protein. So now what they have done, so they use this 32P labeled virus, they infect E. coli cell or bacterial cell with this 32P labeled virus, and they don't wait for 40 minutes or 1 hour. After just few minutes, they remove the viruses which are just stick outside the surface of bacteria, and then they centrifuge it, and they found the bacterial pellet after centrifugation. So, for example, after infection, after few minutes, they just shake the solution vigorously, so that whatever sticks to the surface of the bacteria, that will fall apart, and then they centrifuge. And small viruses, they will be in the supernatant, and bacteria, they are much heavier compared to this T2, so they will be at the bottom. And when they, after centrifugation, they found E. coli cells at the bottom, and they found radioactivity there inside E. coli cell, right? They found radioactivity. But when they use 35S level virus and then they infect E. coli, similarly after few minutes they shake the solution so that remove the virus which is still stick to the surface of bacteria, and then they carry out centrifugation. Then they found E. coli cell at the bottom, but no radioactivity observed here. But in this case, radioactivity was observed. So therefore, it proved that when this T2 infect bacteria and they can multiply, they can make huge amount of fudge virus from the E. coli cell. So there should be some gene, and that is playing this role. But from this experiment, it has been proved that it is when it is the phosphorus is level, that can be stressed inside the bacteria. That means the nucleic acid is entering, the DNA is entering inside the cell, and that causes multiplication of this virus inside the bacterial cell. Whereas when we are labeling, when they have labeled the sulfur, it was not entering, the protein is not entering inside the bacteria. So as a result of that, it definitely proved that DNA is our genetic material. And all these three experiments together, Griffith's experiment followed by Avery, Maglide and McCarthy's experiment, and the last one here, Hershey and Chase experiment, all this experiment, they definitively prove together that DNA is our genetic material. That's all for this class. Thank you very much.